we have one more speaker before the keynote closing, and that's Dan Voytas. And Dan is from the University of Minnesota. He is the director of the Center for Genome Engineering. And Dan has been very successful in using sequence-specific nucleases for genome engineering, specifically in plants, and not only for basic research, but also for improvement of crop. So we're looking forward to hear more about it.
Thank you, Tanya. It's very much a pleasure to be here today and to tell you about the work that we've been engaged in for about the past decade. And our mission has really been to go into living cells and to rewrite the genetic code, to make very specific alterations to DNA sequences. And as Tanya mentioned, this has applications in basic biology. So for the past few days, we've heard a lot of hypotheses that have been generated from sequence generation and analysis. And with the tools of genome engineering, you can go into cells, make those changes, and more specifically, link genotype to phenotype. And then again, the applications of the technology range from correcting human genetic disease, gene therapy, generating microbes or plants that have value for agriculture or industry. And I'll tell you a little bit today about our work in plants, but provide a framework for the technology so that many people here are working on diverse organisms. Hopefully you can leave with a better understanding of the technology and how you might be able to apply it to your specific systems. So the way we go about engineering genomes involves harnessing DNA repair pathways, specifically double-strand break repair pathways. So this slide illustrates a gene target that we're interested in modifying, and we engineer molecules that will go in, recognize that gene specifically in the genome, and introduce a targeted double-strand break. And then we can direct the repair of that break to achieve the type of modification we're interested in. On the right-hand side of the slide, we simply introduce the break and ask the cell to rejoin the broken chromosome. And it often does so precisely, but occasionally small insertions or deletions occur at the break site that can create mutations resulting in loss of gene function. So it's a means of creating gene knockouts. A more powerful application is targeted gene replacement. And here we introduce the break, but we provide a piece of DNA that has homology to our target site. It doesn't have to have 100% homology, however. We can introduce some DNA sequence changes into this so-called repair template, such that when the broken chromosome uses this repair template to copy information into the break site, it incorporates the sequence changes that we're interested in. And these sequence changes can be any number of types. They can be insertions of transgenes. They can be specific nucleotides that will alter the coding sequence of your gene of interest. You can alter the promoter. Any type of variation can be introduced that you're interested in. So the field of genome engineering has advanced quite rapidly over the past few years. And the reason is improvements in our ability to make these targeted double-strand breaks. The field was really launched with a group of sequence-specific nucleases, the zinc finger nucleases, which can be engineered to recognize a specific sequence and create that break. So a typical zinc finger nuclease has an array of zinc finger motifs. Each zinc finger will recognize typically 3 bases. And so you can string zinc fingers together to create a molecule that will recognize your target of interest. And they typically function as dimers. They bring in 2 monomeric nucleases that come together at the target and introduce that break. And this has been a very powerful technology, and it's really sort of laid the framework for genome engineering. But it hasn't really taken off in many academic labs in particular because of the difficulty in engineering these molecules. It takes a lot of time and expertise in understanding how the finger DNA, the zinc finger and the DNA interact. So a few years ago, my lab developed some of the first Tala Factor nucleases, or talons, sort of a next generation of targeted nuclease. And they use a different DNA. The architecture is very similar, but they use a different DNA binding domain, a DNA binding domain that derives from these transcription activator-like effectors, which are produced by plant pathogens. So the pathogen is Xanthomonas, and when Xanthomonas infects a plant, as illustrated here, the bacterium injects into the plant cell these Tala Factors. These are eukaryotic-like transcription factors that make their way into the nuclease, bind DNA, and activate gene expression. And so they've evolved this target specificity to find these specific sites in the plant genome to turn on these genes, which then allow the bacteria to colonize its host. Of course, there's selective pressure for the plant to acquire mutations that prevent binding, and so the DNA binding mechanism has to be very facile and very modular to compete in this arms race between pathogen and plant. So my colleague Adam Bogdanov at Iowa State, I was a faculty member there at the time, and Ulla Bonus's group determined how the Tala Factors bind DNA. And this is an array of repeats in the center part of the molecule. Each of these repeats is 34 amino acids in length, and they're virtually identical with the exception of these two nucleotide, or these two amino acids at positions 12 and 13 that vary. And it's the amino acids at these positions that determine which base the Tala Factor repeat will bind. So if you have HD, you'll bind C, NGT, NIA, or NNG or A. So these four modules allow you to target most any DNA. And each repeat binds one base on the target. And so Adam's group then, along with Barry Stoddard, determined the structure of the Tala Factor. And those 34 amino acid repeats form this little hairpin-like structure. And it's actually just one of those variable amino acid residues that makes a base-specific contact. And as a consequence, this highly elegant and modular DNA binding motif allowed us to engineer these talons. So we could just create arrays of that motif that would recognize the target we're interested in and direct our nucleases to the sites to create those breaks. So that overcame some of the frustration in the engineering of the zinc fingered nucleases. But in the last year and a half, we've hit another big transition in the genome engineering technology, and that's the implementation of this RNA-based DNA targeting. So again, the CRISPR-Cas system originates from host-parasite interactions, in this case in archaea and bacteria. So this is a bacterial cell, and there are loci in the bacterial genome that have collected bits of genetic information from past infections by phage or incoming plasmid DNA. And associated with these arrays of archived sequences are enzymes that carry out an active immunity against future infections by the phage or incoming plasmids. So basically, you transcribe this CRISPR array, and you transcribe and translate the associated Cas locus. You make an RNA that then gets processed and bound by these proteins, and then they are sequence-specific nucleases so that when you have another infection by a phage in this instance, the RNA will pair with it and the nuclease will cleave it and combat the infection. And in the past year and a half, genome engineers have adopted this technology. You can synthesize a guide RNA to your locus of interest, express the Cas nuclease, and very simply and very elegantly, you can introduce a targeted break into your locus of choice. So the frustration that we felt for 10 years in terms of engineering these reagents has basically gone away. Talons were one step forward, but I think by and large, the CRISPR-Cas reagents, because of their simplicity, will be the genome engineering reagent of choice. So in the second part of my talk, I'd like to give some illustrations of how we've used the technology to modify plant genomes. First, talking about really this first pathway, introducing the double-strand break and simply asking, in this case, the plant cell, to repair the break and to recover the instances in which the break has been repaired imprecisely, creating mutations. So the example I'd like to talk about is a practical example, and the goal here was to create healthier soybean oil. So this slide illustrates the major plant oils that are used for human consumption and the percentage of monounsaturated and polyunsaturated fats in these oils. So oils like soybean have fairly high polyunsaturated fats. And so they're typically treated through processes of hydrogenation to remove the double bonds, increase the monounsaturated fats, improve the shelf life of the oil. But as a consequence, you end up introducing trans fatty acids, which have negative health consequences. And so our objective here was to convert soybean oil to a more healthy oil like olive oil that has a higher level of monounsaturated fats. And the gene that we were targeting is a fatty acid desaturase. So this enzyme takes the monounsaturated fat, oleic acid, and adds a double bond, converting it to linoleic acid. So the goal in the seed, the soybean seed, was simply to remove FAD2 activity, fatty acid desaturase 2 activity. So in soybean, like in many plants, enzymes are encoded by multigene families. In the case of soybean, there are two groups of genes, one FAD1A and FAD1B, that are expressed in the seeds. These were our targets because we wanted a seed phenotype. But of course, you need these fatty acids elsewhere in the plant to carry out other functions and other membrane roles. And there are other genes that are expressed in the vegetative tissues. So we engineered, in this case, a couple talons that target sites that are similar or identical between the FAD2-1A and FAD2-1B genes. So these are the target sites that were recognized by those talarays. This is the spacer where the nuclease cuts. There are some polymorphisms between 1A and 1B, but the recognition sites of the talons are identical. So we then introduced these talons. We transformed them stably into soybean. And again, we had 4 targets, the 2 alleles each of 1A and 1B. We regenerated soybean plants that carried the talon, and we anticipated that as those plants grew up, the talon would find the targets, mutagenize them, and then we could recover plants that had mutants either in B, A, or in both genes. And in fact, we didn't really have to make that many transgenic soybean plants. Within 10 transgenic plants, we were able to cover 3 that had mutations, either in 1A, 1B, or 1 in each of the 2 target loci. And then we could take these plants and self them and recover plants that had either the AA homozygous knockout, BB knockout, or the quadruple mutant. And this is the analysis of fatty acid profile from seeds that have these different genotypes. And you can see, again, we were trying to increase the oleic acid level up to that of olive oil, which was approximately 80%. And in the quadruple mutant, where there's no FAD2 activity in the seed, you see that we've accomplished that. And the downstream product, linoleic acid, has been significantly reduced relative to the wild type or the plants that have one functional copy of the gene. So even though it's a very simple target, a single gene target, it can have a profound impact on the phenotype of the plant, and in this case, give a soybean plant that hopefully has a trait of agronomic value. The second example I want to illustrate is one of gene replacement, where we're introducing the break, a repair template to correct the break and introduce a subtle sequence change. And the illustration I'm going to use is one that we published several years ago, but I think it still has quite a bit of relevance, and we're still actually working on aspects of this project. And this is gene replacements that will create herbicide-tolerant plants. So plants that you can spray with a chemical. The chemical will kill the weeds, but the target plant will remain tolerant or resistant to the herbicide. And in terms of U.S. agriculture, herbicide-tolerant crops are widely grown. Soybeans, cotton, corn. By 2009, the vast majority of the U.S. acreage was planted in transgenic plants that were tolerant to the herbicide. So herbicide tolerance was created by introducing, and typically, a bacterial gene into the plant, and then that bacterial gene conferred resistance to the herbicide. Now, most of the common herbicides target genes involved in branch chain or aromatic amino acid biosynthesis. So humans don't encode those enzymes, so the chemicals are not toxic to humans, and so you can spray the upper Midwest with chemicals every year and have very little consequence on animal life, but kill the vast majority of the weeds, save the corn, soybean, that carry these transgenes. So our goal was to create tolerance not by introducing a transgene, but by engineering the native genes in the plant that encode these enzymes for branch chain or aromatic amino acid biosynthesis. The target that we first focused on was acetylactate synthase involved in branched-chain amino acid biosynthesis. And there had been identified a number of mutations, like this W to L mutation at 568, that confer resistance to certain classes of herbicides. So in this study, the talons had not, or the CRISPRs had not yet been invented, and so we were working on the zinc finger nucleases. So we engineered a couple of them that would target this gene. In fact, only one of them really worked well. This one here at site 1. And the way we did the experiment is we introduced a DNA template that encoded this nuclease, a donor that would introduce the modification into, in this case, tobacco protoplasts. And tobacco was our model just because it's so easy to manipulate in the lab. It's easy to make these protoplasts and recover whole plants from these single cells. So in this example of one of the experiments we conducted, we broke the chromosome here, and we introduced the repair template, and we asked this modification some distance, 1,300 base pairs away, to be incorporated into the genome. And in fact, 2.5% of those protoplasts that took up our reagents had undergone this precise modification. Now, as I mentioned, the technology is advanced. We can now use talons or CRISPRs to drop a chromosome break right on the site we want to modify. We can now use single-stranded oligos or very short DNA molecules to obtain at higher efficiency the targeted modifications we're interested in. And then we can take those individually modified cells, regenerate plants, and show that, in fact, they have resistance to our herbicide, so that we've created a trait of interest. So as I said, this is a study that we published a while ago, but we're still working on the technology. We heard this earlier today from Steve about efforts to improve cassava through traditional breeding approaches. And we've been recently funded by the Gates Foundation to introduce some of those same mutations into cassava that would confer resistance to these common chemicals that are being used for weed tolerance or to combat weeds in the US. And so the reason for undertaking this project is weeds in Africa, and particularly in cassava, cause huge yield loss. And so the idea is to increase productivity, but also weeding is carried out by large part by women and children, spending a lot of their time taking care of the cassava fields, not going to school, not being able to perform other duties. So we're launching that project now. And I think as you think about the technology, I drew the comparison to traditional transgenesis, and it sort of brings home the question about how are these new crop plants going to be regulated? They don't have foreign transgenes in their genomes. They're not traditionally GM in the sense that they carry foreign DNA, but clearly they're genetically modified through these approaches of genome engineering. And so it raises the question, if you have a cassava variety that only has 2 nucleotides out of its billions changed, what is the regulatory impact of that in terms of release, growing, and the new technologies demanding consideration by regulatory agencies to address these sorts of questions. So I'd just like to close with, I gave you sort of an overview of the technology and how it can be applied, and I'd just like to close with some thoughts on where we're heading. The 2 examples I gave were single gene modifications. And while they were powerful for introducing traits into plants, the next target is going to be multiplex genome editing, where we edit and alter multiple genes simultaneously to study developmental pathways, to reorchestrate biochemical pathways, to create plants that have even more valuable phenotypes. And these top 2 slides deal with the use of these reagents, these targeting reagents as nucleases, but you can make CRISPR-Cas transcriptional regulators or TAL transcriptional regulators. In fact, the Tala factor itself, the native protein, is a transcriptional activator. So we can use these reagents to create other tools for manipulating genomes and implementing approaches in synthetic biology, altered transcriptional regulators, altered or targeted epigenetic modifiers that lay down methylation marks or create targeted histone modifications. So there's still a lot of opportunity for the technology, and these developments are progressing at a quite rapid pace. So I'd just like to acknowledge the folks in my lab who carried out the work and my funding agencies. The soybean work was done in collaboration with Selectives Plant Sciences. So thank you very much. I'd be happy to answer questions.
Do we have any questions? We have time for a few questions, yes. Sorry.
What's the probability of off-target modification for the CRISPR system? And also, has there been a case where there's been a homozygous knockout of a higher ploidy plants?
Okay, so the first question is the off-targeting of the CRISPRs. So the slide I showed, there was about 20 bases of identity between the guide RNA that directs the nuclease and the target. In point of fact, only about half of those bases are really critical for the targeting. So you have a fairly small sequence signature that's being recognized. And so a lot of the early reports, there was concern about, and some demonstration that, in fact, they would hit other sites in the genome, other related sequences. But the technology is advancing, as I said, very quickly. One of the newer approaches is to use 2 guide RNAs, which would recognize 10 and 10, for example, and then each of them has been created into a nickase. So you nick each strand of the DNA to create that targeted double-strand break. So you can overcome some of these limitations in specificity. And then your second question was, how many targeted knockouts can you make simultaneously, I believe, right?
For homozygous knockouts for a plant that has higher than diploid genome.
Yep. So in potato, which is tetraploid, we've been able to knock out all 4 copies of a target simultaneously. And we didn't worry about it so much in soybean because we could just self it and recover those homozygous. But potato, it's mostly clonally propagated, so we basically had to knock out all copies at once. So it's doable.
We have one more question.
I was wondering if you could do this in a way where you didn't have to have stable tDNA integration. Can you recover the mutations without having to then subsequently segregate away a transgene?
Yeah, no, it's a good question. So in plants, and there's only a handful of them that you can transform and regenerate from protoplast, we can either introduce RNA encoding the talon, for example, or actually purify the talon protein itself. So the mutagen is just the enzyme, basically. And that actually works quite effectively. We can get in excess of 1 to 10% of the cells that have the targeted modification by those means. But of course, the challenge is in plants, many plants, you can't transform and regenerate by that fashion. So, the challenge now is to deliver it by other means. And we and others are thinking about ways to do that, but it should be possible.
Okay, we have one last question right here.
Yeah, Dan, you talked about the regulatory hurdles, and there are 2 sides to it. I guess the US government will have to develop the guidelines, but you as a developer will also have to engage in all kinds of biosafety research to show them that your product is safe. So my 2 questions, what type of biosafety research are you doing, and how much do you think this may cost you?
Yeah, it's a good question. So I'm not a regulator, but we're trying to provide, guidance and data sets. So for example, we have a grant from the USDA specifically to make, in potato, 20 lines of potato modified at the same target with CRISPR-Cas, 20 lines modified with talons, 20 lines modified at that same target by EMS, and then 20 controls. And we're just going to sequence the genome of all of those to sort of get an assessment of collateral damage that these reagents create in the genome. But that doesn't get to your point of every modification is going to be different, And is it going to have, the modified protein elicit an immune response? There's going to have to be a lot of studies that, in fact, will be costly before probably these products can be released. But it's still, those guidelines yet to be written.
Okay, let's give Dan another hand. Thank you.